And the addition of Montmorillonite clay could help speed up this process because the clay was very abundant in early Earth. Whoa. Oh my God. Guys, be on topic. We'll probably have you guys find us in school or something. No idea. Okay. Anyways, so these protocells were self-replicating and can grow in size to absorb RNA and other molecules, which I'll talk about a bit more about this later. Okay. So the first genetic material was self-replicating RNA, or it was very likely to be self-replicating RNA because it not only contained genetic information, but it can also function as a catalyst, which is called a ribozyme when it's a catalyst. So this was because the base pairs on the RNA, since it's single-stranded, they can form bonds within itself and then fold into specific three-dimensional shapes and self-replicate. So, similar to natural selection, the ribozymes that replicated the fastest will be more present and favored. So, any mutations or changes in the nucleotide bases that allow for faster replication of RNA will be retained. And it's not really a contest, it's just we need shirts. Yeah, okay. And as protocells could absorb RNA from the environment, this allowed for further expansion of the cells because hold on, just 1 second. Because higher intracellular concentrations of RNA basically allowed for osmosis of water inside the cells, which causes the cells to expand. Okay, what's your question? Or
So for what are protocells again?
so protocells are basically they're very simple membranes that formed from lipids when they were placed into water. So those were like the first cells because you just have like a membrane layer with a different intracellular and a different extracellular environment.
So they're just membranes. So how can they like reproduce?
So sometimes these the lipids were placed around like certain genetic material or proteins. So those proteins inside, which I just said talked about, they basically increase the water potential difference. So the water will try to flow inside because there's more solute on the inside of the protocell. So that will cause more water to flow in, and the protocell will then expand because there's more water flow. And so when it gets too big, it will have to like split itself, or else like the membrane will start falling apart.
I see.
Yeah. Okay, and as time went on, the RNA could have provided a template for DNA synthesis, which was more preferred because it was stable, because it's double-stranded. Okay. Okay, next is the fossil record. So fossils are preserved rock. Hold on. Okay. They are preserved rock remnants of dead animals, so they mostly accumulate in sedimentary rock, which can be easily packed into different shapes. But some animals can also be preserved in amber, such as like mosquitoes, bugs, whatever. So amber is like tree sap, or ice, including mammoths and some humans. So the fossils aren't actually the bones, the original bones of the animals. They're actually rocks that were packed into the shape. Anyways, it's also important to know that fossils don't the fossil record won't tell the complete history of organisms' evolution since a lot of those organisms won't die in the correct places to be fossilized. Others can be destroyed or some haven't been discovered yet. Okay. Aston, do you have a comment or no? Okay.
I do.
Okay. What is it?
So it's not common actually, it's actually a question.
Okay.
So would cartilage would cartilage be preserved?
Ooh, I'm not sure.
It's not preserved.
I
Because
am not sure because it's like I don't think so. I cannot be sure about it.
If
so I
cartilage is not preserved, then doesn't that mean that
Wait,
that
yeah, if it was preserved though, like noses would also be preserved, and you don't really see those. in
And
fossils.
also, like, the joints would be preserved. Bones are
Yeah.
preserved mostly because they already have a rigid structure, so
Mhm.
they don't like just like fall out or anything. They're not easily decayed.
Yeah.
So
Makes sense.
they survive long enough to be replaced with stone.
Yeah. Thank you. Anthony. Okay. Okay. Anyone else? Chris?
It's like cartilage, and how do they find out like if the cartilage wasn't preserved? How do they
Sorry,
find
you
out this
cut out
element?
kind of.
Oh, for like sharks, I think they're mostly made of like cartilage. So like, how did they figure out there was like a really big megalodon in the back in the days?
Oh man. Oh, I am not sure. If we have time, I'll look it up, but we have like a good amount of stuff to do. We can move on for now. If I remember, I'll like
Okay.
or Anthony, you should look at it. Answer it in the chat, okay? What? What did it say? Yes? So yes to what? Okay. Whatever he said. Okay. Anyways. Did I finish that slide? Oh wait, I did not finish that slide. Okay. Okay, so fossil records can provide a lot of good information, which includes when the organism existed. So this is done through fossil dating or radiometric dating. So radiometric dating is basically based on radioactive decay and elements half-life. So when organisms are living, they will take in nutrients, which includes carbon-14, radioactive carbon-14 and regular carbon-12. So when these organisms die, they won't take in any more new carbon. So the carbon-12 already inside their body will remain stable, so it will remain at a constant concentration in the dead organism. But carbon-14 is radioactive, which means it will start to decay into nitrogen-14. So these radioactive elements have a specific half-life, so it's possible to find out how long ago the organism died by comparing the ratios of carbon-14 to nitrogen-14. So yeah. Also, fossils can provide information about how organisms evolved physically, since there are some similarities and differences between like reptiles here, for example, so and other like mammals later on. So like every one of these has a hinge and like a lower jaw that are very similar. Okay. Here's just like a diagram of all the time periods. So like 4.6 billion years ago, approximately, Earth originated, and then everything started evolving until we got to the human. Cool.
The Homs.
Yeah, okay. And I'll talk a little bit about everything here. So first was the emergence of single-celled organisms. So this was like the longest time ago. So prokaryotes were the first single-celled organisms on Earth, and their existence was basically shown by stromatolites, which are thin layers of rock that form when prokaryotes bind thin layers of sediment together. So as you can see here, the person's like pinching a section of stromatolite, which was formed by the prokaryotes. Okay, next was photosynthesis and the oxygen revolution. So some of these prokaryotes that emerged first were photosynthetic and used light energy to split water, which helped them create their own energy and released oxygen as a byproduct. So a lot of these prokaryotes were water-based, so they just lived in the water, and so the oxygen they released as a waste product dissolved in the water. So at one point, the oxygen got to really, really high concentrations in the water, and so it leaked into the atmosphere. And this high presence of oxygen can be like verified, I guess, with the presence of iron oxide in old rocks. So iron oxide is basically it's rust, so it's red. So this reddish color tends to be in the older rocks, and those have red rust streaks in them. So As the oxygen levels kept going higher, about 2.5 billion years ago, there was an oxygen revolution where oxygen levels suddenly shot up in like 500 million years. So since these prokaryotes were initially largely anaerobic, which meant they weren't using oxygen, these high levels of oxygen, which were toxic waste to them, remember, that inhibited their enzymes and damaged some of their cells. So as a result, prokaryotes either died off or they had to live in anaerobic environments without oxygen, or they had to adapt and start using oxygen in cellular respiration. So that's how cellular respiration started, I guess. Okay. Next was the emergence of the first eukaryotes. So eukaryotes are generally more complex than prokaryotes because they contain nucleuses instead of nucleoids. They have membrane-bound organelles. They have ER and mitochondria, and they also have the ability to engulf a lot of other cells and nutrients and food by changing their shape. So this ability was advantageous since it helped with endosymbiosis when large cells engulfed smaller cells that evolved to become organelles such as the mitochondria and the chloroplast. So after endosymbiosis, the first eukaryotes were formed. So the engulfed cell in endosymbiosis is called the endosymbiont, which coexists in a mutualistic relationship with the larger cells. So the endosymbiont lives in the cell and is protected from the outside environment and other cells that can try to swallow it. And in exchange, it will provide nutrients and energy, such as ATP and sugars for the cell. So another part of endosymbiosis is that mitochondria had been engulfed and involved in endosymbiosis before plastids, such as the chloroplast. And this was basically shown because all eukaryotes will contain mitochondria, but only the photosynthesizing cells will contain plastids. Okay, so endosymbiosis was actually pretty widely supported with evidence. So first of all, the mitochondria and chloroplasts have their own separate division processes. So these mitochondria and chloroplasts divide separately from the cell during cellular division. Also, the endosymbionts have their own machinery and genomes. So as you might have learned or will learn, mitochondria and chloroplasts have their own separate DNA. And also they have separate membranes and transport systems in the endosymbionts that help them with their cellular processes. Okay, so here's just a diagram of endosymbiosis. So first, you had an ancestral prokaryote. So the membranes inside of it did infold from the plasma membrane, which gave it the nucleus and everything. So that engulfed aerobic bacterium, which use oxygen, were able to use oxygen for cellular respiration. And so these bacteria became mitochondria, which helped it use more energy. And then some of the eukaryotes did engulf photosynthetic bacterium that evolved to be chloroplasts. Yeah. Okay. So next is the origin of multicellularity. So the first multicellular organisms were small red algae. And this origin of multicellularity was a really huge change in organism biology because it was basically the shift from single-celled organisms to multicellular organisms. And these multicellular organisms could have had more advantages in helping avoid predation, specialization. So basically, each cell will do its own little thing and have other cells help it do parts that it does not do. And it can also integrate more environmental cues and resources. So later when I might get to protists, there is a kind of protist that will group together to get more food when there's a lack of food. So cool. Okay. Next was the Cambrian explosion. So the Cambrian explosion was basically Austin. Dude, come on, man. Gotta be here to learn bio. Okay, so the Cambrian explosion was the sudden appearance of a lot of animal phyla, which includes sea sponges, cnidarians, which are sea stars and its relatives, and also mollusks, such as this little creature here. So this Cambrian explosion was also marked by the emergence of a lot of larger predatory organisms with adaptations to hunt and self-defend, which includes long spines and body armor. And so prior to this explosion, the organisms were mostly soft-bodied grazers, but these larger predatory organisms had the ability to attack those grazers and also to defend itself. Okay. Do you have a question?
Wait, aren't those called nautiluses?
I'm sorry?
Aren't those called nautiluses?
These on the right?
Yeah.
Sure. But they're mollusks, so okay.
General mollusk. Can I ask a question?
Yeah, sure.
Tell me if I'm wrong, but aren't like cnidarians like jellyfish?
What about crabbies?
Crabs?
Yeah.
I am not sure of
Aston,
crabs.
can you stop interrupting? Can I?
Okay.
Is there a difference between not
So
so
jellyfish are a
these
part of
are
Nigeria.
cnidarians and sea stars are cnidarians, but they're pretty different. I might be asking a stupid question.
No, listen. Yeah, so cnidarians can contain a lot of things. Like we'll talk about later, like protists, that's super general, but that contains a lot of organisms that look very, very different. So there can be like a lot of different things within that. Okay.
Okay.
Okay. Next is the colonization of land. So, during this period, a lot of organisms moved out of water, and this was associated with adaptations that allowed for reproduction out of land without dehydration. So the first organisms were likely to be plants. So they did evolve a waxy cuticle layer on their leaves that prevented excess evaporation of the water. And also plants could have colonized land with fungi because the fungi could form mycorrhizae associations with the plant roots that helped it basically get more nutrients on land. And the most widespread colonizer today is the arthropod, which includes insect spiders, everything like shrimps as well, and also tetrapods, which are four-legged organisms. Okay, so plate tectonics are very important. Yes, what's up? Do you have a question or?
Yeah, I do.
Okay.
So doesn't so because like the wait, let me just first think.
Okay. Do you want me to come back to you or?
Yeah, I just came back.
Okay. I'll just move on for now, but okay. So plate tectonics, the tectonics theory basically said that continents are giant floating pieces of Earth's crust on the hot underlying mantle. So the Earth basically has four different layers, which are the crust, the mantle, the outer core, and the inner core. So the outer core and inner core are the super hot parts, and the mantle has like the biggest volume in the Earth. So regions where the plates slide past each other, so like all of these are plate borders. So these commonly have earthquakes, volcanoes, and mountains. So for example, by the Indian and the Eurasian plate, the Himalayas are about here. And since we live about at the edge of the Pacific plate, we have small minor earthquakes sometimes. And originally, it was a theory that all continents were one big group, which is called Pangea, and this was supported through fossil evidence and species today. So wait, yes, the continents will combine again eventually because they keep moving, because they split apart, so they're gonna like come back together. Yeah, so the division of Pangea caused a lot of species to separate and also the formation of new species once old ones were isolated. And a lot of species were exposed to new environmental conditions. So a lot of fossils that were very similarly related, were found on different plates and different continents. And this was very likely due to this supports Pangea because Otherwise, they would not have been able to cross entire oceans, but the fossils were found in different continents. Okay. Okay, next is mass extinctions. So mass extinctions there's many reasons for that, which can include a drastic change in or destroyed environment. So certain species can go extinct because humans destroy their environment. And so mass extinctions are dramatic changes to environment that can cause a large number of species worldwide to become extinct at the same time. So that is bigger than one single extinction of one single species. So there were 5 big mass extinction events in the history of Earth. So first was the Permian extinction, where 96% of the marine life was altered, and also led to a super active volcanism period. The next was the Cretaceous extinction, which you guys are more familiar with. It eliminated all dinosaurs except for birds, and it was also likely caused by an asteroid or a comet collision. And just something extra, this was supported by the evidence of iridium. So iridium is found in a lot of asteroids and comets outside of the Earth, but it's not found commonly on Earth. But in the places where the asteroid was like what's it called? Where the asteroid was theorized to be land, there was a lot of iridium there. So the consequences of mass extinctions include an altered evolution and ecological community, and also can result in adaptive radiation. Sure. So adaptive radiation is basically a period of evolutionary change when organisms will form new species or to basically take over the ecological niches of like the dead organisms. So this can be worldwide or regional, and basically the survivors of mass extinctions will fill the empty niches to maximize their resources. So for example, if like you and your neighbor have 2 different houses, right? Or if you guys are living in one big like communal house and your neighbor dies, then like you're gonna try to take over their resources as well. And say there's like another person that was also living with you, you would take over your dead neighbor's resources to like minimize your competition with the other person's. I mean, okay, what am I gonna say? Like, am I gonna talk about the finches? It's like a very similar concept. So basically, organisms will try to take over other organisms' niches to like reduce their competition, reduce the already existing competition. So that's seen with the Galapagos finches. So they evolved different beak shapes to try to take over different resources to prevent excessive competition. Yeah. Okay. So developmental genes had changed over time as species also changed. So first, heterochrony is the change in timing of developmental processes. So for example, bats have earlier and longer growth rate of fingers, so they do end up with very long fingers. While humans don't necessarily have the same length of fingers. So also human and chimpanzees have very similar fetus skull shapes, but they have very different developmental processes and timings that are associated with the development of each part that makes their adult faces different. Okay, Aston, do you have a question?
What are axolotls amphibians?
What are Yes. Yes. Yeah. Okay. So, also different species can have reproductive maturity at different rates. So there could be an evolutionary need for this, and certain species have like shorter lifespans or are more hunted and have to reproduce earlier. So for example, certain insects can become sexually mature earlier than bigger organisms such as humans. So next is pedomorphosis, and pedomorphosis basically results from heterochrony, which is basically when an adult form of an organism can contain juvenile characteristic parts. So this happens when the development of reproductive parts happen faster than the other or normal organs. So an example of this is the axolotl, which is a type of salamander. So usually Salamanders don't have gills, but as you can see here, they still retain the gills. Yeah. Okay, and then changes in homeotic genes will also change basic morphology. So the crustacean ancestor had this Hox gene, which gave them a lot of legs. But then nowadays, the same Hox gene did change, and this segment doesn't give them any legs. It's just the abdomen. Okay. Alrighty, next is phylogeny and the tree of life. So, phylogenies basically show evolutionary relationships. So, we name organisms by their genus and species, so that's their scientific name. And species are classified by hierarchy. So the biggest hierarchy, the most general, is domain, goes to kingdom, phylum, class, order, family, genus, and species. Oh, it's okay. So common ancestry can also be shown through phylogeny trees. So this is a phylogeny tree. It's close enough. So but they do branch out like a tree. So a branch point is basically a point of common ancestry. So for example, the badger and the otter have a branch point right here. You can see my mouse. So sister taxons, they share an immediate common ancestor. So badgers, the American badger and the European otter are sister taxon because they share an immediate common ancestor. Rooted trees will show the common ancestor of all taxa on the very left. So the common ancestor of all of these taxa is here at the order. So this is a rooted tree. And also basal taxon will diverge from all the other taxa. So in this example, the leopard is the basal. Austin, can you change your name back, please? Thank you.
Oh wait, sorry for not muting.
You're good. Okay. The basal taxon will diverge from all the other taxa. So in this example, the leopard is the basal taxa because it doesn't have a common ancestry with any one of the other taxa. And it's also important to know that two sister taxa can be rotated at a common ancestor. So basically let's say the coyote and the gray wolf, the coyote could be on the top or the bottom. Like they can be rotated around this branch point and nothing will change because they still have the same common ancestor. I had an example. We don't really have to do it, but it just says like basically which of the trees here depicts an evolutionary history from the other two and explain it. So in this example, C is the tree that is different from the other two because in these trees, basically these are only rotated across the branch points, while this shows a completely different evolutionary history. Yeah, like I said, these sister taxa can be rotated across the branch points and nothing will change about the history. Okay. Next, phylogenies are inferred from morphological and molecular data. So morphological and molecular homologies are physical and genetic similarities because of shared ancestry. So this is different from analogy, which is from convergent evolution. So here's just something to show you the difference between analogy and homologies. So analogy is basically evolving similar features due to like a similar need for that feature. So analogous structure can be streamlined appendages. So sharks, penguins, and dolphins will all have streamlined appendages for swimming, but they are very, very different species. So one is a fish, one's a bird, and one's a mammal, but they have similar features. However, homologous structures are from common ancestry. So all of these four organisms, they have similar features in their arm, I guess. So these don't have to look different. Sorry, they don't have to look the same, but they have similar features. Cool. Okay. And molecular homologies are similarities in genetic material or segments of DNA. Okay. So shared characteristics are used to construct phylogenetic trees. So this is basically done through following cladistics, where common ancestry is the main criteria for placing species and not physical similarities. So species are placed into clades, which so species are placed into clades, which is like different groups basically. It includes a common ancestor and all descendants. Okay. What's your question?
Is that why that even though eels look like snakes, they're not the same species?
Yeah, so I wouldn't say that's due to analogous evolution because um
Eels are fish.
yeah, eels are fish and snakes are snakes. Like they're long because of different reasons. It's not really analogous. They don't really have like the same purpose for being long. Ooh, I don't know. No, I'm very much pretty sure that they weren't, because eels are fish and snakes are reptiles, which have diverged from like a while back. But they do have a common ancestor though, that's for sure. All life does. Okay, so a taxon will only be equal to a clade if it's monophyletic. So if a monophyletic group basically is a clade and it contains of the common ancestor and all of its descendants, a paraphyletic group will contain the common ancestor and some of its descendants, and a polyphyletic group will contain a certain number of the descendants but not the common ancestor. Cool. And then the difference between a shared ancestral character versus a shared derived character is basically a shared ancestral character is a characteristic that originated in a common ancestor of a taxon, which basically separates the taxon from the rest of the phylogenetic tree. So basically, if you see here, the hinged jaw was a shared ancestral characteristic for the bass, frog, turtle, and leopard, while the lamprey and the lancelet did not have that, so these were evolved in the common ancestor. And a shared derived character is evolved specifically in a clade, so those were not evolved from a common ancestor.
What just happened?
What just happened? I don't know what happened. Okay. I'm not sure what happened, but okay. So anyways, the difference between in-group and out-group is basically the in-group is the group we're studying and the out-group is a species from, oh, sorry. Oh, is it because like I moved my mouse up there? Is that what? I think that's why. Okay. Yeah, I think it is. So the out-group is the species that's from, oh, is it fine now?
That's better.
Okay. So the outgroup is the species that's from the same evolutionary lineage as the in-group, but not really closely related, or they don't have the recent common ancestor. So there's two ways to infer phylogenies, which include maximum parsimony, which is basically inferring the tree from most explainable events. So basically the least evolutionary events, and maximum likelihood, which identifies the tree most likely to have produced like a given set of DNA from evolution. Okay. So physical similarities between species can help determine the phylogenetic tree, but ultimately it comes down to molecular similarities, because they can help define evolutionary history that can't obviously be found by physical similarities. So certain DNA, for example, for ribosomal RNA and mitochondrial RNA, they're very similar between all species, so they will evolve pretty slowly. So orthologous genes will result from speciation with a divergence of genes. So for example, here we had an ancestral gene, and after these species were separated, the gene basically diverged into two orthologous genes because each one underwent a little change. And the other type is paralogous genes, which result from gene duplication and divergence. So each little section is like part of the ancestral gene with a little difference in it. Okay, this is something kind of extra, but molecular clocks will help track evolutionary time. So molecular clocks, the idea of it is basically using constant rates of evolution in certain genes to estimate the absolute time of evolutionary change. So here, mutations are plotted against time, and this is usually done with a gene that has a relatively stable rate of change and also a neutral gene, because harmful genes are usually eliminated. And so the mutations will just stop existing because the gene won't exist anymore. Okay, so horizontal gene transfer can impact the genes that get passed on vertically. So for example, the gene for metabolism in yeast, which is a eukaryote, is more similar to the gene in bacteria than archaea, although by lineage, eukaryotes are more related to archaea. And so basically, horizontal gene transfer is when transposable elements like plasmids, viral infection, and fusion of organisms basically take some of the genetic material from one organism and pass it to another one, which changes its gene expression and its genetics. So horizontal gene transfer is really important and prominent because a lot of the prokaryotic genes have moved between species at some point during evolution. Oh, chloroplasts are its own group because they were like their own, they were initially their own photosynthetic bacteria, but eventually they got taken in as organelles. So they are very closely related to cyanobacteria, but they were like brought in as organelles. The bio wrap is tomorrow. Oh my gosh, that was fun.
Cells.
Preston, please mute yourself. Thank you. Okay. Next is chapter 27, which is bacteria and archaea. Okay. Okay. So bacteria have structural and functional adaptations to help them infect cells. So first, they have very specific cell surface structures. So bacteria can have peptidoglycan cell-like structures, which are modified sugars cross-linked by short polypeptides, and this encloses the entire bacterial cell wall. And it has anchor elements that stick out from the bacteria that helps it stick to things and attach to things. So the Gram stain is really effective when diagnosing patients with bacterial infections, and they will help determine the composition of the bacterial cell wall. So if the bacteria is Gram positive, it will have a simple peptidoglycan cell wall like I just described before, like this. If it's Gram negative, it doesn't have that cell wall, and it instead has a complex cell wall which has lipopolysaccharides, which are carbohydrates and lipids. So like I said, these are very important when diagnosing patients with bacterial infections because Gram negative bacteria have lipopolysaccharides, which are more toxic to the body. And they also tend to develop a resistance faster than Gram positive bacteria, since the outer membrane will prevent entry of a lot of drugs. So anyways, the outer sticky layer of the bacteria is called a capsule, and it helps more with sticking to substances and other cells, and can also protect against the host cell's immune system. So next is endospores, which is pretty cool. So to withstand harsh conditions, bacteria can basically produce endospores with their chromosomes. So endospores are basically copies of the bacterial chromosome enclosed in a multi-layer structure. So all the water is removed from the structure, which creates a very resistant spore that contains the bacterial chromosomes. So then the cell will break apart, release these spores, which are super durable and can survive for several centuries before like repopulating other cells. Okay, so next are fimbriae, which are short hair-like structures that help with bacterial adhesion to surfaces. So they are shorter and less in number than pili. So pili are very similar hair-like structures that will help. Oh my God, Aston, stop it. Okay. They're shorter and less number than pili, which help two cells stick together for a gene transfer. Okay. And about half of prokaryotes are capable of taxis, which are movement away or towards a stimulus, which includes light or food or chemicals. So a lot of cells do this with flagella, which are long whip-like structures that propel the cell forward. And these flagella can be on one end or scattered around the cell. So a lot of eukaryotes can also have flagella, but the composition of eukaryotic flagella is really different from prokaryotic flagella with completely unrelated proteins. So fimbriae are usually located like everywhere around the cell because they like help the cell stick to things. Yeah. Okay, Austin.
You just said that what you just like said something that like prokaryotes also have flagella. Is that true?
Prokaryotes?
Yeah.
So eukaryotes can have flagella. Can. Yeah. Cool.
Yeah.
Okay, so rapid reproduction, mutation, and genetic recombination basically gives these prokaryotes a lot of advantages. So bacteria can reproduce really, really quickly, which allows for even faster rates of mutation, like up to 8 million mutations per human host per day. And so this allows for extremely fast adaptation to environments, and this is what can cause your bacterial resistance to antibacterial medications. So even more diversity can be given to these prokaryotes from genetic recombination, which include transformation, transduction, and conjugation. So these are the three methods of genetic recombination, which basically is having more than one organism's genes or more than one types of genes recombined into a new genetic code information. So transformation is when the genetic makeup of a cell is altered by the uptake of external genetic material from the environment. So for example, a bacteria can absorb foreign DNA from its environment and integrate it into its own genetic code. Transduction is where bacteriophages carry genetic material from one bacteria to another. So when these bacteriophages burst out of the cell, they're each of its own little capsids will have they can contain some of the original bacteria's DNA. And then conjugation is where genetic material is transferred between two bacterial cells. So the fertility factor or the F factor will determine the ability to form sex pili to interact with other cells. So if you are F positive, then you can form these pili. If you are an F negative, you cannot form these pili and you can only be on the receiving end. So here, as you can see, the F pili, the pilus, will allow for transfer of genes through the sex pili. So another important factor is the R factor or the resistance factor. And this allows for resistance to certain types of drugs. So if you have these factors, you can spread them around. So that's another way how drug resistance spreads, basically. Okay. Aston, do you have something to say?
So if if the first bacteria landed on Earth, what does that mean that it came from somewhere else?
No, the first bacteria, we were talking about this in the first chapter.
Sorry.
It's okay. So bacteria cells are prokaryotes, which are the first types of cells to originate from Earth. So these cells were protocells that formed. So the first cells were protocells that formed by themselves, and so they started to expand, be able to self-replicate, and those first cells were prokaryotes. Okay. So anyways, an example of a drug, guys, be nice, okay? An example of a drug is an antibiotic drug is penicillin. So that can kill bacteria. But
Never mind. Never
No,
mind.
I was answering the question. So those that it can kill bacteria, but bacteria often have very strong resistance to that. So stronger and stronger antibiotics have needed to be formed. And so there was the threat of like a superbug that's resistant to all antibiotics. That's pretty dangerous. So yeah.
I think I take penicillin. It looks a lot like the medication I'm taking right now.
Are you taking it for bacterial infections?
I don't think so. I don't know. It looks a lot like it. Like the other.
Okay. How does it look like? It's usually like a pill. Like a
Yeah, it's
capsule.
just like it's just divided in
Okay.
half in the middle.
Yeah. There's a lot of other types of antibiotics too, but I don't think penicillin's like prescribed super commonly if you want to take it orally because like there's it's not prescribed super orally because like I said, there's a lot of resistance to that already. Okay, anyways, this is hold on. Okay. This is transduction. Sorry about that. Where basically the bacteriophage will infect the cell, and once they break out, some of the new bacteria can have a little part of the a little part of the original bacteria's DNA, and then that will get transferred on into other bacterial cells, which then incorporate that into its DNA. This is conjugation, where an F plasmid, so an F positive cell that is the donor cell, it has an ability to form the mating bridge or the sex pili, and then it can like basically split its plasmid into two and then like have like replication of a new plasmid, and then that's how it gets transferred. So oh, by the way, plasmids are like little circles of independent genetic material in the bacteria cell that can get transferred. Okay. Wow. So impressive. Okay. Next is nutritional and metabolic adaptations in prokaryotes. So yes, what is?
So does that mean that eukaryotes can have sex?
This is in prokaryotes. There is something kind of similar in a type of protist. Like they can exchange their nuclei. They can fuse their nuclei basically. Aston asked that. Okay. So in oxygen metabolism, there are obligate aerobes that have to use oxygen for cellular respiration, and also obligate anaerobes that cannot use oxygen for cellular respiration or they will die. So these obligate anaerobes will live by fermentation or using anaerobic respiration, which basically involves extracting oxygen from substances that contain oxygen, such as like nitrate, or sulfate, which both have oxygen in them. So another type of metabolism is nitrogen metabolism. So some species can convert atmospheric nitrogen, which is N2 dinitrate, into its dinitrogen, into its usable forms, which is called nitrogen fixation. So these bacterial cells can group together to cooperate in metabolism and form compounds that the other cannot. So an example of this is the Anabaena. It has genes for both photosynthetic and nitrogen fixing cells. So some of these cells can only fix nitrogen, which are heterocysts, and others can only photosynthesize. So when they live together, both of these cell types can benefit, and it results in biofilms that can recruit other cells to join and grow their colonies. So here's a heterocyst. It can only fix nitrogen, and these are photosynthetic cells that produce energy. Okay, so there are a lot of bacteria in archaea. So bacteria include proteobacteria, chlamydia, which are parasites that have to have a host, spirochetes, cyanobacteria, which are the most closely related to chloroplasts. They use photosynthesis, and some have components for nitrogen fixation, like the bacteria we just talked about. And also gram-positive bacteria, which is its own little thing. So gram-positive bacteria can also cause a lot of diseases, and these include mycoplasma, which are extremely tiny bacteria without a cell wall, and these are extremely hard to kill. And also archaea. So archaea are eukaryotes, but they're not like eukaryotes per se, but they are a separate domain from bacteria. So they include the extremophiles, which love to live in extreme environments, and that includes the halophiles, which lives in very salty environments, and thermophiles, which live in very hot environments. Guys, can you like chill in the chat, please? Thank you. And also methanogens, which produce methane waste when oxidizing H2 with carbon dioxide. Okay, prokaryotes have a huge role in the biosphere. So certain prokaryotes can act as decomposers for chemical recycling. So they can convert molecules. They can convert like locked up molecules and nutrients into usable nutrients for other organisms. And this includes nitrogen fixation and productions of sugars and oxygen. Certain prokaryotes can also immobilize nutrients in soil by turning them into unusable forms. So there is a very complex relationship with soil and plant nutrients. And also symbiosis, which includes mutualism, which is seen here. The bacteria can produce light for the fish, which attracts prey. And commensalism, where the bacteria can benefit and the organism does not really get affected. And also parasitism, where the bacteria will benefit and take nutrients from the host organism, which is then put at a disadvantage. Okay, I need to go fast because this is our last like chapter. Okay. So prokaryotes can have very significant effects on humans, which includes disease. So there are mutualistic bacteria that live in our gut and can help to digest components that we can't, and also produce carbohydrates and vitamins. They can also stimulate the human cell to secrete certain antibacterial components that targets their competition in the gut. There are also pathogenic bacteria that cause disease by release of poisons, which include exotoxins, which are proteins extruded by pathogens, and also endotoxins, which are lipopolysaccharide components of the gram-negative bacterial cell walls when they break down. And horizontal transfer can also make harmless bacteria harmful. So essentially, before E. coli was harmless, but strains that cause bloody diarrhea have emerged from horizontal transfer. And also bacterial defense systems, we have studied those like CRISPR-Cas9, and that has helped change genetic expression in the genetic components. Yes, Marco.
What is gRNA?
gRNA is guide RNA. So that is produced from the external genetic material that comes in, and that helps the enzymes recognize new foreign DNA that comes in. If it has the same sequence, it will target that sequence, cut it out, basically cut up the DNA, and so it can't like harm the cell.
What is E. coli?
E. coli is a type of bacteria.
Like what? Is it a virus?
It's a bacteria.
Oh,
yeah. We're only talking about bacteria today. And then lastly, bioremediation is when prokaryotes are used to remove harmful substances from the environment and produce nutrients instead. Okay, we can stop here. I had another lesson, but it's too long. So I think we can post that. Post the slides for you guys to like read over. It's about protists. Just like memorize the protists, know some of their characteristics. Yeah, cool.